יפה בקשר מדהים. כן, אני אשמח, אני פה עולה עליו. לא יודע, אבל משתפר פה, משתפר. כרגע משתפר, יואב.
קובלנו 19 מעלות, אבל יש פה 25 כאלה.
מה מראה 19 מעלות?
קובלנו 19, אבל יש פה 25.
לא, אני לוקח את זה בניסון דקות. שלום, אהלן ירדן, פרופסור ירון.
לידי יש פה כיסא של
מקומות קבועים זה חשוב, מירב.
I'll be fine.
תמונה מסעי. This is life.
אבל הוא טוב את Hi everyone. Good afternoon everyone, nice to be back after this long break. And we're happy to have today Cohen with us from Weizmann Institute. And then he's assistant professor at Weizmann. After doing his PhD there with Ronnie Palvin and Anne Steinman, he moved to Boston, he worked in the MGH at Harvard. And then his past 5 years, 4 or 5 years, he's back at the Weizmann Institute. His lab works on canaries and bird songs and canaries, and the idea that the song can be parallel to human speech, basically by choosing different sequences of operation of syllables. He had an interesting, I forgot the notes that I wrote. He had an interesting paper from 2020 where they discovered that there are hidden states in the HVCs of canaries that can actually encode latent states of the song or the previous part of the songs. And today I think we're going to hear about how the songs evolved with time, but not only the syllables, but also how the changes between the syllables and the transition matrix between different syllables evolve during learning. I hope it was close enough.
You can.
Okay.
Thank you very much.
Thanks. But it's really kind, and it's important to emphasize, it feels almost strange to be back in normality. So it's a pleasure, and thank you so much for inviting me. I'll do my best. Yeah, so the lab, Cohen, HVC, and Weizmann, our main impetus and question is, we want to know how does the brain compose. So we study the neural mechanisms that underlie syntax control, the way you control the ordering of elements in our behavior. Today I'm going to talk to you, and I'll try a story on you because it's a work in progress. I'm going to try about how we dissect the plasticity of the syllable transition mechanisms in the canary premotor brain nucleus HVC. Yes, so actually many of our behaviors are sequences of motor or vocal elements. And in extreme examples, like the ones I'm showing you here, we have experts who actually train quite a lot in their lives to produce sequences. Here's Glenn Gould, the pianist. He is a master, and he trained, and he can produce a precise sequence of music notes. And here is Simone Biles, the gymnast, who does whatever heavenly magic she is doing, and she has trained a lot, so she's able to replicate, almost in a very stereotyped manner, the sequence of her, gymnast actions, basically. Okay, but these are, of course, extreme examples. Us normal mortals, our behavioral sequences are much more variable and rich, and we create large repertoires of behavioral sequences that obey ordering of syntactic rules. And this is what I mean by that, just ordering rules. And let me just give you an example. So we can think of a sequence of words in this example. Let's say canaries like to. And then I can choose to say maybe sing, maybe fly, with equal probabilities. But if I change the first word in the sentence, let's say neuroscientists, then I will be more likely to say neuroscientists like to sing. So we have a transition here whose probabilities depend on what happened several steps back in the sequence. This is an effect or a manifestation of a syntactic rule. Okay, this is of course just an example for our main question in the lab. So how does the brain control the syntax or the ordering rules in the rich repertoires of behavior sequences? We want to know how each one of the action, or what are the neural activity states that drive each behavioral action, and what are called transition mechanisms that move from one action to the next and actually produce the ordering of the syntax rules. And we want to study it in an animal model that is, or a behavior that is used for social interaction, and it's also a learned behavior. So we study birdsong. Now, if you stumble into a birdsong talk, you are most likely going to hear about this bird. This is the most studied bird. This is the workhorse of songbird neuroscience. The zebra finch, they're quite lovely. And what they're specialized to do is to learn a sequence of syllables, a single sequence from their usually fathers as juveniles. This is a sequence of about 4 to 7 syllables that last about 1 to 1.5 seconds that they will repeat again and again throughout their lives, which can be as many as 7 years of life. And I will show you that there are also underlined in a few slides that these repeating motifs are underlined by a repeating sequence of premotor neural states. So if you wish, these birds are the Simone Biles of birds, this one version. There's other birds currently being studied. These are, for example, Bengalese finches. They have a more flexible or variable sequencing options. And so there's a transition diagram between different syllable types. There's a few points of flexibility here. However, it's quite difficult to separate the timescale of the syllables and the syllable transitions. So we chose to actually study a different bird, the canary. So here's an example. Right, this is not even a full canary song, it's just a short snippet. And yeah, below the bird I displayed the sonogram or spectrogram of its audio. This is the time and frequency, and the repeating markings here are syllables, we call them syllables. And this is a short snippet. A full canary song can be between 10 to 20, 30 seconds typically. Expert canaries or excellent canaries can go over a minute, but it's rare. If I give each one of the syllable types a letter, like we did here, then typical canary songs will look like this. Okay? The longer or the typical length ones that are a few tens of seconds basically never repeat exactly. It's almost impossible to find two that are exactly the same. But you see a lot of structure here. You see different syllable types, marked with different letters, have transitions. We can build a transition diagram like we did with like they do with the Bengalese finches. So this is a transition diagram of one canary. We need to collect quite a lot of data to do this. You see that their songs are strictly hierarchical. Every syllable repeats a number of times. It's not the same number of times every time the bird sings. We'll talk about it in a minute. And we call these subsequences phrases. And the sequencing of phrases has the same, let's call it memory-dependent transitions or syntactic dependencies, right? We have transitions between phrase types, so one phrase transition to another, that depend on what happened a few transitions or a few seconds earlier in the sequence. This is a bit like the example I showed you from speech. So canary songs are very rich repertoires of hierarchical and flexible sensory model sequences that are learned. So juvenile canaries, like all songbirds, learn the syllable types from first the fathers usually and other males in their vicinity. We don't know enough about actually the rules, if they learn those as well or not. It's not unknown. They're used for communication. These are mate attraction songs usually. Right, males sing to compete with each other and attract the females. And specifically for canaries, and for example not for finches, their songs are also relearned annually in this seasonal cycle where they will pick up new elements to their song in autumn in preparation for the next spring season, mating season. And this is underlined by a lot of neurogenesis in the premotor areas that drive song. Okay, so today I'm going to show a little bit of our experimental access to this animal model, to its behavior and to its neural activity. And we will build the analysis of this on what was studied in actually zebra finches. Again, I told you this is the workhorse of birdsong neuroscience. People could really leverage the benefit in these birds because they have this repeating motif. You can record neurons in their brains and time align it to exact time point in the song motif in which it was active. And people recorded this is a sagittal slice of the finch brain, and I highlighted two brain nuclei. This is the premotor pathway. This is exclusively, as far as we know, exclusively responsible for learning and driving of song. This is the premotor pathway. It's homologous to the motor cortex control of motor neurons, basically in the spine, or the brainstem. So this is the nuclei HVC and RA, and when researchers inserted electrodes into this nucleus HVC and recorded individual neurons that are active and project to this next station RA, they found something quite striking. These neurons are active at specific time points in the song motif and nowhere else, right? This is the time point in the song motif, and the y-axis I'm separating, first of all, it's not my figure, it's from a famous paper. So different colors will be, ticks in different colors will be spikes of different neurons, and they're separated by the different neurons and also by repeats of the song motif. So every time the bird sang the song motif, this neuron in greens fired here, very reliably and very robustly. And other neurons did the same, but in different time points with about 1 millisecond, 2 millisecond precision. Okay? If you record in the next premotor stage, in this nucleus RA, you'll find neurons that again are firing very reliably, time aligned to the song motif, but in multiple time points in the song motif. And this is what we think drives the precise motor output of these birds, because the next station after RA is the motor neurons, okay, in the brainstem in this case. So this is what drives the precise muscle activity of these birds. And for our working model, I'm going to package this stereotype activity that drives stereotype behavior into what I'll call the neural state or premotor program model. So each syllable will be marked by a box, right? And we need to remember that there's this precise activity of neurons, let's say at the level of HVC, that drives this, the production of this syllable. Okay, so it also means that if I record one neuron in HVC and it's active and I hear this syllable, I can accept that the entire premotor state is being produced. Okay, this will be our working model. It allows for us, for example, to think about how sequences are being generated, because Now we can let one premotor state either repeat or drive the next one. So you can create syllable repeats, and we can also create stereotype sequences. We can also create branching points, points where the one syllable can lead to multiple others. In my past work, I showed that in the canary brain, there's other premotor neural states that can drive the same syllables by different neurons in different contexts. That is what the concept. So in this case, this allows, for example, for the canaries to produce these in different sequences following different contexts in the song, and therefore relay the memory, the working memory, of what happened here to a transition point here and create this history-dependent transition. Okay? Today, I'm going to show you how we leverage some of these properties when you want to say something about how transitions, or the plasticity of transitions. Okay? And we're going to try to compete or to compare two different hypotheses about how these transitions, probabilities in this case, can actually change. When we think, remember that these are underlined by the activity of neurons during the production of one syllable, which transitions to the activity of neurons in one of, in this case, two different syllables. And this can...
Can I ask you something? Or can I ask you
Whenever, yeah, of course, questions are welcome.
So can you
Mid-sentence, yeah. Say it again?
one slide back?
Yeah, of course.
I wonder, I meant two back, I think.
Okay.
In between. Okay, this one. So if I look at the upper one and I look at the lower one, and you say, well, it's syntax because the transition is whatever, depends on earlier history. But at least in this drawing, it's completely two different sequences.
Right.
So once you're here, you're there, so it's already determined by the time, right? I mean, so it's, I don't know if I call it syntax, I call it one syntax, one sequence, and another sequence.
Right.
So it's
If I hear this syllable, right?
Right, but what I'm saying is that the way it's implemented in this example, but maybe it's just an example, is that these are two completely different series. So there's one set, they happen to share few items,
Right.
but it's not like you online manipulate something as I would think about syntax, right?
So it includes this branching point where you online manipulate a transition. We think this happens online because, as far as we know, these are the same regardless of the history, for example. So you have branching out and branching in transition, and this will allow you, for example, for this syllable to be produced after this context or after this context. So it is flexible.
You mean there are two different types of...
The other option would be, sorry to interrupt, but the other option would be that the bird needs to include a single sequence for every possible song it might produce, and these are more songs than neurons at some point. I'm, of course, joking, but...
Wait, so just to summarize, right, so if you have a single motif, or a note or a two-note, okay, right, so the idea is that motif is represented by a single neural state, right, so even if you have a...
By motif, you mean a sequence of syllables or just one of them?
Let's say a syllable, right? So a syllable can appear in different phrases, but that syllable will have the same neural representation irrespective of the sequence that it's in.
So that's the point here, and this is not the point of the talk, this is just to tell you something about the history, is that no, I mean, in zebra finches, yes, but in these birds, you can produce the same syllable. Again, this is defined by us, but as far as we can define it, the same syllable can be produced by different neurons in HVC, exclusively different.
Okay.
And this allows for the birds to encode something else, but in this case, we think it's the context in sound. Okay, but this is not the point of the talk. But okay, we're going to talk actually about transitions today, and about the fact that they can actually change. In finches it doesn't happen, in canaries it does. I'll show you in a few slides. But let's think about what can happen mechanistically. So one option, right, we're talking about neurons active during the production of one syllable and then transitioning to neurons active in other syllables. This can change, the probability of transitions can change, if, for example, the activity of neurons here change. This will create different initial conditions to whatever mechanism takes us to the next step. And this will result in different transition likelihoods. At the same time, or alternatively, we can think about an option when this doesn't change. But if these are synapses, then there will be a change here. Okay, so the connections, the strength of connections changes, and therefore the likelihood of transitions will change. And let's see if we can at all tease apart these options. Maybe not. But start with the fact that these things actually change. So this is work of Sivan in my lab, an alumnus of the lab. And she looked at different transition points, so points of transitioning between syllable A to B, C, or D, and followed them, measured them across days, and found that in some cases, for a certain syllable A, the probability to go to B, C, or D will not change across days. This is the timing. But in other transition points, different syllable A's, there will be the change. Okay, so this changes. And actually in about 30 to 40 different transition points of each bird's repertoire. But it's not the only thing that changes. Actually, at every description level of song, and I'll show you a few examples, you find changes at the time scales of days, of across days. So for example, we talked about repeats, right? I showed you a sonogram, and I showed you that syllables repeat. And then because it's a phrase, and the number of syllables per phrase is not fixed, so we can look at the distribution, or the histogram, of how many times the syllable repeated, and we do that across days. So every row here is a histogram, and for a specific syllable type, in this case, of one bird, across days. And in this case, it is stable. In the same bird, a different syllable will have a histogram of a different syllable that actually changes across days. Okay? It does. We can look at the syllable acoustics, right? We define it by a point in a 7-dimensional space of acoustic features. One of them can be the duration of the syllable, one could be its fundamental frequency, and other properties. But every time a bird produces a syllable, it's a point in this space. So we can look at the cloud of points for a specific syllable type of all of the renditions from a single day. It will be a cloud here, and other days this cloud will move and change its shape. So we measure two things. We measure the distance between clouds of pairs of days, and also the changes in variability, acoustic variability. And we do that by changing the volume, by measuring the volume of these clouds, which will represent for us the acoustic variability. And so we can measure changes in variability by changes in the volume of these clouds.
These clouds are the same syllable?
For a specific syllable type.
So each point is like an average of the production of that syllable per day?
No, it's one rendition per day.
Oh.
Take all the renditions and we build a cloud.
So a cloud is a single syllable per
Yeah.
day?
Yeah. Okay. And we can look at, for example, this distance as a function of the distance between days for a specific syllable type. So every point here will be a comparison of 2 days, and we see strong correlations in about half of the syllables. This is just one example. And we can do the same thing with the changes in variability. So the y-axis here will be the change in cloud volume as a function of the difference between days. And as days are apart in some syllables, so does the variability change. Some go high, some go low, but this is an example of an increase. Yes?
Is the change in a specific direction?
So, no. In some it goes high, in some it goes low. Different story from this one. I'm not going to talk about it today, but if we look at all the features together per individual bird, for 4 out of 5 birds we looked, they mostly go all in the same direction. So birds tend to globally get more organized or less organized. This is not a story. Okay, so we have changes in multiple levels of description, right? So we're in a bit of a trouble, right? How can we use it or rescue from it? It could be that these things are actually connected with each other, right? And we have a hint about these might be actually connected from a work done in Bengalese finches in the lab of Michael Brainerd. And he looked, or they looked, at the acoustic properties of syllables, marked here by these letters, right before a branching transition. So you can have a sequence leading to transition to A, and the same syllables leading to transition to B. It's called a divergent transition. There's also convergent transition, which is just the opposite. And what they did is compare the acoustic features of the same syllable H in the two contexts. What they found, it's a bit of a difficult slide, but the gist of it is that the distance between syllable types, or between those clouds I just showed you, is greater 1 point before the transition and lower as you go back, approximately. Okay? But it means that there is a relation. The changes, the variability between acoustics of the syllables here can actually predict maybe the variability of transitions here. Okay, we can try to measure it, right? So that's what we do. We look at the acoustic variability of a syllable A, all renditions of syllable A, we look at the cloud, we look at the volume, okay, or the change in volume, and we look at the variability of the next transition. So this will be, I'll call it syntactic variability, but about what I mean really is the entropy of the transition probabilities here. Okay, yeah.
How different does a syllable need to be from itself to be considered a whole new syllable? How different does a syllable need to be from itself to be considered a whole new syllable?
Mostly, because canaries produce syllables in repeats, we take all syllable, all those that are in the repeat and call them by the same name. They're clearly distinct from the adjacent phrases, but it's a valid question because in some syllable types to be very similar to each other, and this is experimenter dependent. Some people tend to oversplit, some tend to overlap, but this is done over and over. In what's called expert annotators, but in 95% of the cases, it's a clear cut, right? The debates are always on the phrases. Okay, I hope this is okay. So we measure this, right? We measure the acoustic variability of this syllable and the transition variability or the entropy of the transition probabilities and their changes across days. So here are two examples that had the same correlation, magnitude, right? We correlate the changes in acoustic variability with the change in syntactic variability, right? The change in this syllable's variability and the next transition variability. And in both cases, we have a significant correlation. And these are for two different syllables. Every point here is just a comparison of 2 days. So we can color the points by the distance between days as well. And here we see a difference. This is the case where it didn't matter. The colors are spread throughout the figure. And here there is a correlation between all 3 variables. A change in time, or across days, a change in acoustic variability, and a change in syntactic variability. So we can use partial correlations, discount the effect of the acoustics on the days and the transition variability, and see maybe the changes in acoustics of syllables actually explain away the change in syntactic variability. And it's not the case. So in 5 different birds, we see a significant fraction of its transition points marked by the stem syllable. That actually, after removing the effect of acoustics, we still see a significant correlation between the time, the change in days, and the change in syntactic variability. So maybe we can somehow dissociate 2 different things. The premotor, or pre-transition premotor state, which is here represented by the acoustic of a syllable, and the transition selection mechanism, whatever that is. Okay? We try to simulate this, see how this can work, right? This is a work of Nadav in my lab and other rotation students. Not shown here, I'm sorry. But we model this selection mechanism using an extension of the SynFire chain. If it's not known to people here, anybody here did not hear about the SynFire chain? I need to explain it. No brave people. Everybody knows.
It's a generational thing, I think.
Hmm?
It's a generational kind of knowledge.
It's age-related.
Everybody knows.
No,
That the boat is leaking.
the opposite.
The
The
captain lied.
does, no.
Okay. I'll explain it anyway because I don't see a consensus. It's a work I think starting with Moshe Alvarez some years ago. If you think about groups of neurons being active at the same time, this will be the time axis abusing every notation here. So these will be groups of neurons active at the same time synchronously, and they will be fully connected to the next group of neurons that receive synchronous inputs, and therefore they produce synchronous outputs, and this synchrony progresses to the next neuron, etc. But in this case, we have a branching chain. This chain branches into 2 other options. They compete. I'll show you in a moment how. And one of them wins. So we have a branching transition. So we can use this to model syllable A, syllable B, syllable C, and think about the transitions between A to B or C. Of course, this is a model. We don't know where syllable A actually ends. Maybe it ends here. And this is part of the transition mechanism. We don't know. Okay? This is unknown. But we need to model this. We can change parameters about the probability and strength of connections between those states. And we add inhibitory neurons to also have them compete with each other. Okay? So maybe, right? Maybe we can model one of them and fail to model another one. Right? Maybe. Right? Let's try. Let's try to see if we cannot do this one. Well, actually, so what we need to do is to change the synaptic strength in these connections. We do that, and sure enough, we can change the transition probabilities. But the x-axis here will be strength in these synapses. The y-axis will be probabilities of transitions between A, in this case to C, which is the red line, and to B is the green line, and to nothing at the end of song. Right? This happens too, right? In the blue line. So we can model any combination basically like this. Okay, so we cannot rule this out, right? Okay, maybe you can rule out this one, right? Better. So this will be modeled by inputs to this state. This can be thalamic inputs. HVC is part of thalamo-cortical loops. Loops, several loops, so never mind. So it gets a thalamic input. It also gets auditory input from the homolog of auditory cortex. Birds don't have cortex, if anybody wants. So let's try doing this. Also works. I mean, we can change this input strength to some of these, selectively to some of these neurons. So random, but preferring one option, and increase one probability and decrease the other, just like we did here. So we cannot do this. Okay.
So I'm not sure I understand that option that you just presented. So you have neural states that represent syllables, right? So you're saying that something happens with those neurons that would change the transition to the next syllable.
Right. So two options, right? It could be that between days, right? The synapses. The synapses here changes their strength. Okay.
But to the next syllable, right?
And
That's like what,
that's
like
okay.
the standard thing. Yeah.
Okay. Another option, and this has been done when people try to modulate the adaptation of birds to repeated syllables, to explain the distribution of the number of repeats. They did that, so they changed the acoustic, basically response, or the auditory response, and has an input directly to this nucleus HVC. That auditory part of the brain, never mind their name, we have a direct input to this nucleus HVC. This can adapt, these synapses can adapt. This will affect the strength of the input, right? So if this input changes...
So that's so it affects the...
Yeah, we change the inputs to these neurons.
Okay, that's weird. I mean, at least the way that I think about it, it's an odd hypothesis, right? Because A is just, that's the thing that tells me I do this phrase, right? And so you're saying that to change the transition to the next phrase, somehow I change.
Yeah, effectively I change the initial condition to whatever goes on here. So this actually affects them.
Yeah, I mean, it can, yeah. I understand it. It's just not the way that I normally think about it, but okay.
I mean, so these are sensory motor processes. I mean, it's not only, I mean, you can deafen birds and they will sing, it's true, but but they also respond to, I mean, neurons in HVC also have auditory response. And there's also thalamic inputs. Okay. Okay, so we do this and we cannot rule out this option. Okay, so let's go and ask the neurons, maybe they know. Okay, what we did is to recode neural activity in these HVC projection neurons, the ones project right outside of HVC. We did that by implanting miniscopes, miniaturized fluorescence microscopes. People here use them, I hope, but if anybody who doesn't, everybody has even theoreticians have fluorescence microscopes in the lab, I know. Those are really big things, right? But the mini ones are about this big. They weigh about 2 grams, and birds can carry them on their heads. And enough of them are happy enough to sing. And so we can, with a fluorescent microscope, we make cells in HVC express GCAM, or fluorescent calcium indicator. And you'll see here the activity of cells. It will be color-coded. This is a processed video. Don't be surprised or alarmed. It's not real colors. as this bird that doesn't have a miniscope, it's a mock bird, sings, and below you'll see the annotated syllables of these birds, of this bird. Just an example. Okay. So if we go back to the working model and remember that every time we hear a syllable produced and we see a cell in HVC active, we can imagine the entire neural state being active. You see here the orchestra in this bird's brain that plays its notes, basically. So we can look at the activity of these cells and try to contrast these options. And because we use miniscopes, we can actually track the same cells across multiple days. So in this case, we track them between 4 to about 28 days. But here's an example of a cell that was tracked for 19 days. There's not no 19 boxes here, but it's tracked for 19 days, right? We can identify the same neuron, and it's active exclusively in the same phrase throughout that time. So we can look at its signal level at that phrase across all the days and collect enough data, right? What you see here is a raster. Rows are songs. This is time in the song. The green line is the onset of a specific phrase type, just one phrase type. And the red line is the end of that phrase. And we collect signal, we call it S. And we also look at what was the next transition outcome. We call that Y. Okay? Okay, so what you see here on the right is basically that if we collect everything together, is what's the probabilities of transitioning from the fixed phrase type here and every different type of the next option, what I called earlier, I called them B, C, D, and so on. Okay. Can we use that kind of data to rule out, let's say, this option? Let's try. Okay. So what we do is we take this data and we break it into days. And we can measure the probabilities of transitions of what Y is in every day. So this will be x-axis here. And we have here cumulative histograms, right? Or distributions, right here. But per day. So in the first day, this was one transition option, which was less likely. Now it's more likely here. So again, we know there's changes between days. So this is the data, the behavior data. How can we rule out the option of no changes in a transition selection mechanism when all the effect is in the actual pre-transition state? First, we asked, is there any information in the signal here to what is the likelihood of the next transition? Yes, there is. I'm not going to show this, but it's the quantified information. It's in about 40 to 50% of the cells we encode them. What I will ask now is, can we use this, right, if there is information, and imagine there is a stationary probability of finding outcomes of transition Y if we know what is the signal. This is, for me, it tests the hypothesis that there is no change or a stationary relation between the current state and the next. Okay, the transition itself doesn't change. All that changes from one song to the next is just the signal before the transition. Is that clear enough? Okay. So we model this, right? We use a few components. We use two likelihoods or two distributions, one in strong signal cases and one in weak signal cases. And we build a mixture model of them using a mixture function, alpha, which is only dependent on the signal. Okay? It's a sigmoid. Okay? And when we do that, we get perfect fits. We can predict, it fits these distribution perfectly in all cells and all transition points. But can we use this to actually predict the per day distribution? It was fit on all of the data. Now we can take the signals from individual days and fit them and see if we can predict this. And in this case, this is a yes. Yes, we can. In this case, in this cell and this example, we compare per day the predicted distribution to the measured one for uncorrected, and cannot find any pairs where there is a significant difference. Here's another example of a different cell, little colors, where the likelihoods change not in a monotonous way, but sort of a sinusoidal way, if you wish. And again, we didn't find any significant differences between the measured distributions and the predicted distributions. However, we do find cases when there is a difference. Here's two examples, two different cells, barrage of colors to see if anybody is still awake. Very good. And this is two cases when we can find significant differences between the measured distribution of transition outcomes and the distribution that is predicted if we assume that all the changes between days is the signaling of cells before the transition, but the transition mechanism doesn't change. That all the change is the initial conditions of transitions. And this happens for about 30% of the cases. And if you only look at transitions that actually did change across days, because not all transitions change across days, it's about 50%. Okay? So if we wanted to rule out this option, right? We saw that we have information in the signal here about the outcome, so we cannot rule out this option, but we also cannot rule out this option. In fact, what we see is that both options, or of both parts of the mechanism, can be dissociated and both change in the time scale of days. Okay? And of course, we try to make this more difficult and control for different options. For example, in passing, I showed you that the phrases can be of different length, right? Different number of repeats. So we can control for this and see that still we get the same effect. This is the boring control. The more interesting one is actually trying to make this model a bit more complicated, right? I told you this is a sigmoid as the function of the signal of one cell, but you can actually include multiple cells. There's a few occasions when you can have pairs or triplets of cells recorded in the same phrase type. So instead of taking just the signal of one neuron, we take a vector of the signal of a few neurons, and so we have the sigmoid with these coefficients. And we can also add the acoustics of syllables as another reporter of premotor activity. So this would be this vector A for every syllable, but in this case it's an average per phrase, or median per phrase, but per phrase. And we can do that, so here is just rasters for how pairs and triplets look like. It's, we color-coded two different cells in two different colors here. It's activity time aligned to the onset of a specific phrase type across multiple repeats of the same phrase in many songs. And this is the same idea, the outcome of the next transition. This is pairs of cells. This is just colorful pictures, of course, but you can see that there's differences between the rows, right? Cells did not signal in the same way every time the bird was singing, and the outcomes are also not the same, but we can repeat the same exercise. We can fit the model, perfectly predict the transition outcome likelihood, and see if it predicts per day, and see if we have discrepancies. And in every case, we find discrepancies, so we also include the acoustics, right? Of individual syllable, the same idea. We take the 7-dimensional acoustic representation of a syllable, or in this case per phrase, and include it in the model. And in both cases, we find that, yes, there is a significant fraction of the pairs or triplets where we cannot rule out the transition mechanism plasticity. And it's especially high if you only look at transitions that actually change across days, because if they don't change, this is in parentheses here. If they don't change, of course, it's highly unlikely to find anything. We did one last control. Of course, this is also doesn't help us. We did one last control. We look at cells that are active specifically right before the next transition, right? So this is, again, the raster of activity to a specific cell, and it's time aligned not to the onset of the phrase, but to the offset of the phrase. Right before the next transition, and even sometimes going over it into the next transition, and still, in these type of cases, in a significant fraction of the cases, we cannot rule out the plasticity of the next transition mechanism. Yes?
I want to make sure that I understand. In the case where you do see that the state-dependent transitions change, one interpretation could be this is an indication that there is plasticity. There must be plasticity. Another option could be that you're just not measuring the state fully. You see some of the cells.
Right, exactly.
So do you have an answer to
So
this?
I'm showing you here our best effort, right? We include as many cells as we can measure, and we also include the acoustics of the syllables being produced, which is the reporter of the the best, we're not radical behaviorists. Still, it's the best reporter that we have. HVC has 100,000 cells. We don't record them all, we record in individual phrase types or syllable types, we record 3, 4, right? Best.
A conceptually related question is, could it be that your model is not good enough? Or your mapping is not accurate enough? Maybe with more non-linearity would have worked?
So we don't want to overfit. So what we do is only compare or fit the global distribution or condition likelihoods. With the simplest. But, I mean, if you, for example, took a...
The fact that you are unable to explain the change and the transition does not mean that there's no information, full information in the system about this change. Maybe you just didn't find the right rule or
always
the
the
right
case,
technique.
of course. Also, unobserved things like asked here or unaccounted for variables. Again, I am fishing for any, of course, suggestions and whatever reviewers will say, because again, you can complicate things until you hit the statistics wall. For example, I briefly glossed over it, but we took one number for every neuron for every trial, but it has a custom response shape. We can account and take more numbers from every trial. Again, until we cannot fit models anymore, right? This is what we did so far. Okay. Sorry. Okay. So we can't rule this out for now. Yeah, and controls and other complications. Again, in each of these cases, we made a very large fraction of cases where we cannot rule out the option that the actual transition selection mechanism is plastic across the time scale, if you wish. Okay, so let's summarize what I showed you. It was a bit dense. Let's see what you say. So I showed you that in canaries, which is unlike other songbirds that are studied currently in systems neuroscience, as far as I know. I'm happy to hear other data. We have transition points where the likelihood of transitions change across in the time scale of days. Okay? Although the acoustic properties of syllables change across days. This could be problematic, but we actually used it, leveraged that both of them change, to try to tease apart two options. The difference between changes in acoustics of syllables and the changes in the variability of the next transition. We did that using partial correlations and found that we can tease apart the pre-transition behavior and the transition behavior in many of the transition points in 5 different birds. We then asked if we can actually figure this out in a simulation, and so that we can simulate both mechanisms. One mechanism is where transition outcome likelihoods change because we impacted the pre-transition state, and another option where we actually change the plasticity or the synaptic strength to the next state. And then we asked the neurons, and we saw that, yes, we cannot rule out, cannot contradict the option or rule out the hypothesis that both mechanisms change in the time scales of days. So we remain with this conclusion, that we have two mechanisms that seem to be acting not independently, but not 100% connected, right? They can be dissociated, and they both change in the time scale of days. And maybe to pitch something that is hip right now, this is an example of what you do when you go into natural neuroscience. You let the animals express their natural behavior. Yeah? You look at them doing what they do in their day-to-day. And in this case, if you let these two animals run, sometimes they run at the same speed, right? They're dynamic, but they're stationary. You can imagine that they have the same mechanism, right? But when you allow them to express their natural behavior and change their behavior, right? One of them accelerates much faster, and the bird wins. and thank you very much. This is people in the lab. You saw mostly the work of Kenan and Nadav, and Sivan is already an alumnus. We have funding. We have many thanks to all of you. My father took this picture. Another case where the bird wins. Yeah, questions?
Yeah, I just have a question about the conclusion. So, if I understood you correctly, you did manage to account for a lot of the variability in the transition by capturing the neural state, which was only represented by 3 to 4 cells. So, isn't it more safe to assume that by capturing hundreds simultaneously, you would account for a very large, percentage of the variability in the transition, and not that there's plasticity in the way? I don't know.
We started with the what's accepted in the field that these states are stereotyped, right? I tried to break it, so I don't know how much I can break it, but that's we're heading, right? With miniscopes, we can track cells across multiple days, but within the production of each syllable type, they're very sparse, the cells that we can see. We can record about 100 cells best. And we think many more contribute. We're doing electrophysiological recordings these days, but we will not be able to track cells across days, so many days. Yeah.
So is it possible to imagine a feasible experiment that would give a clear answer?
Apart from recording every cell.
Well,
so what you do is try to.
could imagine thought experiments, but
Yeah,
I don't know
so
that you can do that, that it's feasible. If you could have access to the state of each synapse.
What we will be trying to do is to impact things, right? To do a perturbation experiment. So we actually started doing these things, and this may be an answer.
What if you decouple it from the sum? Could you just take the neural activity and then make some hierarchical structure?
Right.
Ask what the traditions.
So this is a specific case. It's an excellent model because it's active exclusively in sum, except in recent years we have works where you see the same cells being active when the birds are asleep. We do record neural activity in sleeping birds. I'm not showing it here now, but in electrophysiology. And we do see replays, maybe.
No, but even if it's doing songs, and then if you do the partitioning not according to syllables, but just according to some algorithm to try to get different parts of the speech and then ask a condition between them that are not conditioned on the syllable itself. That will. It's unlike it's like a control.
Use the neural activity to actually pass
it only
without knowing the behavior, and then
it might not be interesting, but you want to see if you get something stationary, or you also have to change it between days.
I feel like this will be much more feasible with what we do with electrophysiology. We do have cells that can be tracked across these pairs of days, and then we have also inhibitory neurons that are much more active. The property of these excitatory projection neurons, it gives us one burst of spikes. It's very stereotyped, and we've replicated this with electrophysiology in HVC canaries, and just in cells that we just cannot track across 20 days. I'll get to everybody. There's that. Yes.
Yeah, so when do you do the experiment with the,
Sorry?
when the experiment are performed, all of us, so during the year, at which period of time?
So these are seasonal singers, they sing in spring. We keep 2 colonies of birds, one is in the flipped season, so we have them also singing in autumn. I don't want students to get too lazy. So we have about 2 periods of peak singing for about 4 months.
Does it change
So these were recordings all during the spring season, around April and May.
So that's the time was it changed?
It's a time when they're supposed to be the most stable and produce their showtime songs. The breeding season. They're actually a lot more variable in plastic in autumn. Right? Yeah. This is springtime recordings.
Yeah.
Yes, please.
So this talk was titled about plasticity, but couldn't this be framed in terms of drift, right? Because there isn't, as far as I can tell, there isn't a particular task where they're supposed to change the transition from this syllable to that syllable, right? It just sort of happens that instead of saying this one, they sing that one. So, there are changes.
What drifts?
Like representational drift, like just the either the like your two hypotheses, right? Either the representation of the syllable itself changes, or the transition changes. But it's not necessarily due to some sort of like directed plastic mechanism, the way that you think of say some sort of supervised mechanism, or let's say some sort of Hebbian thing to stabilize things. Right. This is just stuff that's changing.
Let's separate two things that you said.
Yeah.
One is like supervised. I have a goal, right?
Right.
I have a teacher, and the Hebbian plasticity can happen without a teacher, right?
Right. So
two different things.
supervised and unsupervised.
And you can imagine this happening,
neither.
right?
This is neither.
Why?
In other words, I don't know which one it is, but this seems to be something where instead of, because the process that you're describing is a process of destabilizing something. Right, you have a stable pattern, and now you're changing it to something else. So this is almost like a process that could potentially be due to increasing some sort of noise in the system. Right, this isn't something where it's trying to stabilize something or toward something. It's trying to change things.
You're casting a wide net. So I did not dwell on this, but in some of the birds, they actually shift throughout, say, 2-3 weeks of time that we record them, into more stable behavior. Stable being all the variability of multiple features of song that I just described is reduced. If you think about several repeats, the histogram will become narrower. It will be more stereotypical. Every time they will sing the same phrase, this number of repeats will be similar. If you think about transitions, reduction in entropy, meaning they will prefer one outcome more than the others in a stronger way and become more stereotyped. This happens as well. For now, we don't know if we're catching them in different phases of something that can be periodic or in a directed process. We don't know. We don't have a perturbation experiment, which we are starting to carry this season, where we actually try to reward one direction versus another.
Right, because to me it seems like those, definitely, right, I'm a musician, right, and I write songs, right, so there's definitely a period where I'm trying to experiment and do different things. There's a period where, I'm going to perform in a week, so we have to practice and get it down. And those are very different processes, right, so I think that you have to disambiguate behaviorally what they're trying to accomplish.
Right.
And because those are very different things, right.
You're tapping into an active research directly in my lab, but it's not the topic of the talk, and I want to add other people as well. Yes. I feel the angry eyes. Yes.
My question is about terminology, so it doesn't affect any specific thing, any of your messages. But why grammar? Why grammar?
Oh, did not say grammar.
Syntax.
And I make very clear as well. Yeah, so that's important. And I get this. I find in the beginning, it's for the sake of this talk, we call the ordering rules. I call them syntax rules. It's easier. It's part of what syntax means in speech, but it's not all of it. It's true.
It's almost none of it.
No, it's not. That's not true.
I try to argue that at least if you think of human language, if I wanted to say something about syntax, I would do my best to try to separate it from semantic, context-related effects. So here, I imagine a sentence, and in the sentence there's a bird, and in the end there is fly or say. There's an association between bird, the word is semantics of bird and singing and flying and so on. And it has nothing to do with the order of words specifically. It's simply the fact that in the context of birds, sing and fly are very likely, right? And in the context of men or women, flying is not so likely. But that's not what this in linguistics would be called syntax.
100%. And that's
Why is the word syntax? It's just the fact that I think when you use this kind of terminology that comes with a lot of baggage,
right.
It sees confusion rather than clarity.
I have to agree with you, because it does, but in some cases it really helps, I'm sure. And a more serious answer, and again, this is speaking a bit of a debate, but people here are interested. I think it's a real debate, and you see here models of language that are completely statistical, have no, and all they learn
so they have no syntax.
and all they learn is probabilities, and they produce a text that follows syntactic rules. And who is to say that you're not doing the same? So I think it's an open debate. This definition does not comply with all of the linguists' definitions. It's correct.
So the transitions are so rich, I'm wondering whether some of them mean something behaviorally.
Right.
Because in the zebra finch, when you do it in reverse and things like that, then it has an impact on the behavior. It maybe relates to what Willem is saying.
Yes.
And so I'm saying behaviorally, can you identify that the transitions are meaningful?
Right now I can't. We are actually running these experiments. What we do is video tracking of motion and vocalization in birds that either hear playback of other birds or actually are in interaction with other birds. And we're trying to decipher how what is their responses. David is still here, so I cannot diss the French. But it's an important body of work, but it was done starting in the 1990s and early 2000s about how female canaries respond to the song that they hear, and it's very sparse signal because they were only looking at specific body postures called copulation solicitation displays that occur preferentially when they hear specific syllable types. But we think this is extreme cases. And actually, they respond all the time when they hear, and you want to actually analyze their behavior, the ongoing motion, and see how they respond to what they hear. So hopefully, within a year or two, I'll be having much less mumbling answers to your question. I'm sorry for that.
Just a mechanistic question: are they alone in the cage, or?
Yes, they are.
So they don't hear other birds.
They don't.
Thank you.
Thank you.
סיימנו כזה וננסה להגיע לכל הסינטקס מסודלת כנגד ההחלטה.
גם לכנראה בסינטקס מולה.
נשמח מאוד לשמוע, הוא ישמח לשמוע את זה.
באתי להגיד שלום, וכל הזמן רצית משהו, וראה
לא, אני באמצע העבודה. אתה יכול ללמוד סינטקס מתוך זה. אתה יכול ללמוד סינטקס מתוך זה. נקבע בגין שלך. זה ויכוח אחר של הסדר של המילים יש סדר כאן אתה בסדר זה המילה הכריזית שלך זה בסוף של בנים כל מה טרנזישן קשור לסמנטיקס
קשור לסימטריה.
זה במאה אחוז. אני צודקת,
ההפך, אני
עשיתי מאמצים לעזור לשפה.
עד שזה היה יותר מדי, ואז התחלתי לדבר פליק, אבל הפרובביליטי הזה
אבל זה בשמות הזה. אחד אליהו, ממש
יש לי 8 בבית, אתה חושבים בבית.
לא, ב10 בבוקר אני לא מציע.
אבל מה שרציתי אתה יכולת לקחת סוף מילים בשפה, להציג אותו בסדר נורמלי, בתנאי שהמילה,
זה היה קצת
פה, שזה לא במשפט, כלומר, חסר סינטקס של חלוטין.
וואו,
אני יודע
אתה צודק, אבל הוא עומד לי גם.
זה תמיד, אני...
למה לא יכול
זה
בדיוק זה,
הסיטינג זה דבר,
יש לו מטרה.
יש לו קונטקסט סמנטי, יש קונטקסט רמתי, אתה למשל יכול להגיד דברים שסמנטית וגם מופרחים, וסינטקטים וגם מופרחים, ולגיסטים יש כל מיני דוגמאות מצוות כאלה. זה גם נכונות, עולה להם שוב סמנטית, יש קונטקסט כזה, יש קונטקסט כזה, זה לא אותו דבר. צריך להסביר אחרת.
נכון, זה כן אומר, תשמעו, אני מסתכל פה שתלויים בהיסטוריה, זה דוגמה וזה במנותק, זה סנסור לא במנותק, אבל אנשים תמיד יגידו, זה לא מה שאתה מרוב אפשריים, וזה נכון.
בתוך הדיבייט, איך אתה לומד, איך נוצר סינטקס, הנה תראו, אני אומר לכם שפשוט מתוך הסדר הזה אתה יכול ללמוד טרנזישן פרובביליטיז, יש לך מספיק, אז בסוף אתה תלמד גם שכל נסבע אחרי או לפני הכל, אז זה כן יעשו, אז זה כן יעשו, שכן יעשו,
אז אני אגיד לך, איפה זה עוד נפרד?
איך לך קשה ואחר כך 5.
לא, אבל צריך להגיד לו, לדרוש בסוף שהוא
זה חכם, כאילו לא משהו שיש לי אינדיבידואל.
אם לא אומרים,
התכונה של הקבוצה.
אני ראיתי את זה אחרי
ופה אנחנו אפילו מנסים להבין
רק עכשיו
מהי השפה של הקבוצה.
אני פעמיים באתי באיזשהו
בי של
שבוע פעמיים
לא, יש לנו שפה משותפת, אבל יש לנו אידיוסינקרסיס. אבל לאינדיבידואל, שוגים באופן שוגים, הם לא שוגים. באופן קבוע במין ובמספר של צמיג. זאת אומרת, זה שאני חוקר על תכונות של האינדיבידואל. ולכן אני פעמיים חוטא להסדרה של הבלשנים. כן, לא, אני לא יודע להגיד לך את הילדים שיש... אז יש עוד דבר אחד שבאמת
שזה הגיע לכאן, פרסם ברבים.
החזרה, זה נולד. אם אתה לוקח את הנריס, ואתה מאמן אותו על שירים שהם יותר לאמן
תחזוקה,
הם משחזרים שירים שהם חזרות. הם לומדים את זה, וגם אתה יודע שבחיים לא שמע חזרות. כשהוא מגיע לעונת כזו ושמע שלו, הוא יקח חלק מהמרות שמגיעים ויתחיל לרצה לתת. זה מולד.
כל הזמן מציג.
תודה רבה רבה.
אז אני כל הזמן אני מציג. ולפעמים זה עובד. אז אני מציג כל פעם, עד שאני מתעסק.
הנה, נפנה לך את הדרך. היה נייס, very nice.
תודה רבה. ניסיתי משהו קשור, אני לא יודע איך להציג את זה.
זה קשור.
איך להציג משהו על דרך
תציג את זה עוד 3-4 פעמים, אנחנו נבין.
לא, זה בסדר. אני רוצה מסוגל לכתוב את זה כמאמר, אז אני רוצה לדעת איך אני יכול... אם זה בסדר.
זה טוב, ולפני שכתבים כמאמר זה מאוד טוב. לנסות על אנשים ולראות מה תופס אותם ומה פחות תופס.
להראות משהו שהוא לא, כן?
אבל אני חושבת באמת השאלה, does it matter? באיזושהי צורה זה צריך להיות איזה משהו פונקציה, האם יש לזה איזשהו
אנחנו עושים את הניסויים, אנחנו באמת...
אתה קורא לזה למידה. למידה זה לא סתם change, זה למידה, זה כאילו... קראת למידה?
אמרתי בהתחלה שספרים...
נדיר.
פלסטיות. האם זה במטרה?
הפוליטיקלי קורקט.
אתה זהיר, בסדר.
אומרת מה שאני יכול למדוד ולהראות, כן? השאלה היא של האם... אבל השאלה של האם אולי בכלל שהייתם, לא הקלטתי, שאלה קבועה לכולם, כן?
אבל הייתה לי עוד שאלה עקרונית.
אני רק רוצה להגיד תודה.
תודה רבה. אם יש לכם באמת מחשבות קדימה, אני אשמח לפידבק. אנחנו כותבים, אבל אתה יודע, who knows, כן? תודה.
כותבים זה לא מצב נקודתי. כן,
נכון, טוב איתה זה לא נגמר.
אני אגיד לך, התחיל לכתוב.
מזל שיש לי קביעות, הוא אין לו קביעות.
ואז שלח אותי ליונתן.
אני לא שלחתי אותך.
יונתן, זה כמו משהו בור בלי תחתית.
זה היה אירוע מזוכיסטי שאת יצרת לעצמך.
אז אמרתי, אביאל, די, לא, אולי ככה, אביאל, מיצינו.
אביאל יש יותר כוס וגם לך, וואו.
זה מלחמת...
כן, אביאל הוא כזה...
באמת אני מאוד אוהב אותו.
הוא נגן כינור. האלה, הנגנים האלה, הם כאילו... ממשיכים לעבוד על איזה מין... אני לא בטוח, יש פה עוד איזה משהו שלא בדקנו, ועוד משהו שלא בדקנו. ואתה מכיר את ההצהרות עם האפטר זה ואפטר זה, וזה לא סימטרי? הנה השאלה שלי.
כן.
כאילו אתה מדבר...
תודה לך לצהריים, תודה.
אם... אתה מדבר על זה כשתי אפשרויות לכאורה בלתי תלויות. זה הרבה likely להיות תלויות, נכון?
בדיוק.
לא, זה מה שרציתי לשאול אותך, למה? Why don't you embrace them both? למה
לא,
אתה שם אותם במלחמה?
אני כל הזמן... כן.
אני embrace them both בסוף, כי אני מנסה לשבור. ואני אומר, תראו, יש פה שינוי.
אתה אומר, אני לא הצלחתי לי שאני רוצה לשבור.
למה לראות את זה ככישלון?
בדיוק.
הפרייז, איך אומרים?
פריימינג,
זה מה שרציתי להגיד. הוא יכול להיות...
זה גם וגם, כאילו זה גם זה וגם זה משפיע.
כן. אני כל הזמן יש לי אסוציאציה בתור מישהו עם איזה קליני כאילו, אז אני חושבת אנשים הם יותר לייקלי, בסקאלה של חיים, נכון? יש לך גנים שעשו לך יותר מעלה לסקיצופרניה, סתם. אבל, depending on the context, השאלה אם תלך לפה או לפה, זאת אומרת, יש לך איזה predispposition מסוים, ועכשיו השאלה, זה שני דברים שהם הביחד, והשאלה אם אתה יכול מהביחד.
זה ככה, נכון, את צודקת, כן, אבל השורה התחתונה צריכה להיות, יש פה יותר ממנגנון איחוד אחד, אלא שניים, והם לפחות, והם שניהם משתנים.
לא, זה בסדר.
ואני לא יכול כוח להפריד ביניהם.
לא, זה בסדר.
אבל לא ברור לי שהם בלתי תלויים.
נכון, הם לא בלתי תלויים.
אז עוד יותר.
זה טוב שהם לא בלתי תלויים.
נכון.
זה נכון שאתה צריך פרמטר, אני גם חושבת, תכף שנגיד, אני חושבת כמה זה נחמד אם היה לנו הכל פרמטר אחד. בתור חוקר, אבל אנחנו חוקרים את המוח של עצמנו. תאר לך שאת המוח של עצמנו אפשר היה להסביר בעזרת פרמטר אחד, זה אסון נוראי.
כן, G.
אז אנחנו יותר מסובכים.
G? ה-IQ?
נכון, בשביל זה יש את הסושיאל.
כן, מבנה אחר, פחות יפה מזה.
זה נכון שאפשר הרבה להסביר, a long way מפה, בעזרת קצת. והווריאביליטי, כמו הנגנים, הווריאביליטי בסוף זה קופץ לפה, אבל זה קפיצה. גם בסוף הרי אנחנו רגישים למשהו שונה, נכון? אני כאילו, מישהו שהוא בערך בגובה שלי, מה זה רגישה? אני צריכה טיפה להרים את העיניים, אני צריכה... ומישהו שהוא פה. כאילו, יש תחום שבו אנחנו מאוד רגישים, ועוד תחום של השונות. רוב הדברים בינינו הם מאוד דומים בסך הכל.
כן, מכירה את טוני זיידר, יש לו את הסיפור הזה עכשיו על למידה, שהוא אומר, אין באמת למידה, זה פלאף, כאילו רוב
כל נתון,
הפלסטיטי זה בכלל נלמד באבולוציה, זה עושה טיונינג לפרמטרים קצת.
ברור, אבל זה מה שחשוב לנו, הטיונינג הזה, כי that's what we have. זה ה100% שלנו.
טוב, אני חושב שאנחנו נפגשים עכשיו.
כן, אהלן, רגע, אני לא ששארתי שום דבר, קטנות לילדים.
אני כל פעם שאני נוסע לסמינר אני מאבד ציוד.
כן.
יצאתם מהזום, מהחשבון?
לא יודע מה יהיו.
הנה, חשבון של מישהו.
לא שלי.
אני חושב, אנחנו לא שמים בזום.
person. So you arrived in Israel?
I came yesterday.
Wow. Okay. But the jet lag from India is not.
It's not that bad. This class is okay. The last class is okay.
Yes, the last class will be late for you.
Yeah, that'll be closer to midnight.
Yes. Okay, but good for me. I forgot my glasses.
It's all very quiet here.
I'm sorry?
So it's all very quiet here.
Yes?
In Israel. I'm just-
In Israel. Quiet. I mean, yes, for the last, I don't know, week or two. You know, we just don't know. It's also a bit strange because, you know, one day missiles are in the air and the next day we are like normal. So is the shifts are it's not easy to adapt yourself. I mean, everyone are a little bit confused, like, so what? So now everything is normal? It's like,
So let's continue to be normal. It looks normal, let it continue to be normal.
yes, but we remember that we were in a very unnormal place for a relatively long time. I mean, it wasn't like a week. I remember reminding myself that actually I also last year, in the last lesson or two in the semester, we did it in Zoom because it was the other war, I don't remember what it was. So it's like for already like a long time that things are not normal. But okay, I mean, this is it. Okay, let's see. So you live here in Israel?
In Jerusalem, in student village. So.
You came here for your MBA?
Yes.
Nice.